Hey guys, how you doing? So welcome to chapter 13. We're going to look at the second video, and in this video it's just going to be a short kind of history lesson on genes and some of the scientists that have made major contributions to the discovery or the understanding of genes. Okay, let's jump into it. Okay, so we're going to look at a few people, three people in particular. Okay, Gregor Mendel and his work on peas in 1856, and Frederick Griffith and bacteria in 1928, and Oswald Avery and enzymes in 1943. We're going to look at how each of these people have contributed to our understanding of genes and genetic inheritance, and why their contribution is so significant. Okay, we're going to look at a few other things as well, but we'll go from there. All right, let's start with Gregor Mendel. So, born in 1822 to 1884, he was a monk. He was a monk at St. Thomas Monastery in the Czech Republic, and he conducted experiment on peas. Yeah. He recorded parent plants, crossbred them over many generations, and he kept records of these plant lineages, yeah, which at the time was quite a rare thing for him to do, or for people to do in general. And he managed to actually isolate very specific traits about these peas, yeah. And this is what he did, yeah. He was so careful and so meticulous about the process, it was almost painful to think about it in that kind of way. He bred well over 500 plants, and each one he would grab a part of or a portion of the flower, and he removes the stamen from them. He would then collect the pollen with a brush, and then he would actually pollinate other flowers by hand using this brush. So it was by doing that he was controlling in his experimentation, controlling the environment in which the plants were reproducing, and by doing that he was able to then select for very distinct or certain traits within his peas. And so that's what this table or this diagram here kind of shows, right? Mendel managed to develop two variations from plants, yeah? The stem length, he managed to isolate tall stems from short stems. He was even managed to isolate certain colors. If he wanted a yellow pea pod, sorry, not a pea pod, a pea color versus a green pod. The shape, it was either round or wrinkled. The coat color with either gray or white. The pod texture could be inflated or constricted, green or yellow, and even the flower position could be either axial or terminal, right? And so it was quite interesting the way that he was able to very specifically isolate some of these traits. And there was a reason, and he was able to come up with some really cool explanations in that, right? So Mendel discovered that each trait was controlled by a pair of inherited factors, yeah? For example, he named one of them as seed color, and the factors for affecting seed color was either green or yellow. You could have two of these options for seed color, and one of the seed color would be green and another one would be yellow, and he was able to isolate those two particular traits. Yeah, they could be identical. In other words, if he managed to get a long and long, he called those like a stem length, long and long, he would call them purebreds. If it was long and short, he would call them a hybrid, right? And he discovered that in those characteristics, some of them were what we call dominant, and others were recessive. In other words, the ones that were dominant seemed to always take the lead whenever he crossbred two types together. So if he crossbred, for example, a green seed with a yellow seed, he found that the yellow seed always came up, in the offspring, whereas the green one weren't. And so the yellow were the dominant, and then the green were the recessive, right? And during gamete production, one member of each factor was separated into different gametes. So he kind of the first understanding of parent genetics being different from offspring genetics or gametes, yeah. Now, his work was largely ignored, unfortunately, because they didn't think he was qualifications. He was a monk doing scientific work, and the scientific community at the time didn't really accept it as being qualified work, and so they sort of just brushed it to the side, just thought, oh yeah, it was just this, this monk's ramblings and about these discoveries. However, only in the 1990s, his work was rediscovered and became widely recognized, and still is today. Yeah, so he's recognized as the kind of father of genetics, and the whole understanding of genes originated from his observations. Okay, now if you think about it, in 1900, how old was he then? Yeah, I'll leave you guys to think about it. Right. Our next guy, Frederick Griffith, born in 1877 to 1941. who was a British biologist, and he conducted experiments with bacteria that caused pneumonia. So pneumococci, I can't remember how to pronounce that, right? Bacteria in his experiments. He found that there were two strains of bacteria. There was one with this smooth strain, which if you looked carefully, they had this capsule outside the cell wall, right? And it would cause pneumonia. There was a second strain that was a harmless strain, which was a rough strain, in which case the external capsule, that one that was outside the cell wall, was absent. It wasn't there. And those bacteria were harmless, right? Now, Frederick Griffith was a total jerk to mice. He performed, tons of experiments in which he killed like large populations of mice, probably, through his experimentation. So what he would do is he would inject living smooth types into the mice, which resulted in their death because it caused pneumonia, and he just sort of watched them die, I guess. And then he also injected living rough types into mice, but they lived. Yeah, he injected dead smooth types into mice, and they lived. And then he would do this combination where he injected dead smooth types alongside the living rough types, and they died, right? And so here is a diagram to show sort of what happened, right? You can see there he had a very good control mechanism there, right? Living smooth cells means death, right? So it was the smooth cells that was causing pneumonia and killing the mice. The rough ones were harmless because he injected the living ones and nothing went wrong, right? If you killed the smooth cells, however, the mice would also live. But if you killed the smooth cells and you put them together with living rough cells, both which independently caused no change in the mice beforehand, the mice would die from pneumonia. So what he was observing was that something inside these dead smooth cells was causing the living rough cells to become deadly. Yeah, and so he didn't know what it was. He couldn't figure out what it was. So he called it this transforming factor. Yeah, that there was something in the dead cells that was causing the living cells to kill the mice, even though the living cells previously were harmless, right? And it had to be something that was non-living because the dead smooth cells were already dead, right? And there was nothing in the living smooth cells beforehand that was killing the mice. And so it had to be some sort of chemical substance that was creating this kind of deadly factor, killing the mice, right? He never really figured out what it was, and no one did until along came a guy named Oswald Avery, right? Born in 1877 to 1955, American physician and medical researcher, right? And in 1943, make note of the date, he discovered the chemical nature of this transforming. He figured out what this transforming factor was, and he did it by extracting the smooth type bacteria and took that capsule that was encasing the cell wall, and he treated it with enzymes. Yeah, enzymes that would destroy lipids, enzymes that would destroy carbohydrates, proteins, and nucleic acid, right? And he figured out that the nucleic acids were the enzymes that destroyed nucleic acids would make the bacteria harmless again, right? And so, oh, by the way, it was worth noting, how old was Frederick Griffith during this time in 1943? Yeah. So what he discovered, the transforming factor was a nucleic acid of some sort, right? Because if he treated them with protein-destroying enzymes, it still was deadly. If he treated it with lipid-destroying enzymes, it was still deadly. Same with the carbohydrates. But then, if he treated it with DNA-destroying enzymes, the transformation ability of those rough bacteria disappeared, and they couldn't kill the mice anymore, right? And so this was the first conclusion that genes were actually made up of DNA. Yeah? And so that was the nucleic acid aspect of DNA, right? Once again, his discovery wasn't immediately accepted by the scientific community, and it was only after 1960, 20 years later, right, did his theory of genes become widely accepted, and he was kind of more, recognized for all his hard work. Now, 20 years later, how old was Oswald then? If you did the maths with all three scientists, you probably would have figured out that all three of them had died before their recognition was given to their work, right? And so, I like to think about it, sometimes your greatest achievements are pretty much not recognized until after you're dead, right? So, it's a big thing even in science, and, in Hebrews 11:39 actually says, they were all commended for their faith, yet none of them had received what is promised. Yeah, to understand that kind of big race that we are all running in the process. Okay. All right, let's hop into the definition of genes, and then we'll finish it up there. Okay, so in 1911, right, they defined the genes as a name for a thing in a germ cell that makes the germ cell develop a particular characteristic, such as tallness as opposed to dwarfness. Very basic understanding. Right. 1921, hypothetical unit in a germ cell that determines the production of a particular characteristic in the individual derived from the germ cell. Still also a hypothetical unit. You can see their very brief definition. Right. 1934, still a hypothetical unit in the chromatin. Now they discovered exactly, the strand that the gene was in, right, that had a specific influence on certain characteristic. And then we have in 1966, after Watson and Crick were able to discover the structure of DNA, a segment of DNA molecule that can copy itself and pass on to other generations and directions it contains. Right? And now in 2011, we have a locatable region of genomic sequence corresponding to a unit of inheritance that is associated with regulatory regions, transcribed regions, and other functional sequence regions. Basically, it is a unit of inheritance. Okay, so we'll stop it there, and then in the next lesson, we're actually going to look at how genes work and how they code for genetic instructions. Okay, see you guys.